Vanakkam. Welcome to NCERT Biology Reading. NCERT Class 11, Chapter 9, Biomolecules, 9.7 Structure of Proteins. Proteins, as mentioned earlier, are heteropolymers containing string of amino acid. Structure of molecules means different things in different contexts. In inorganic chemistry, the structure invariably referred to the molecular formula. Example: sodium chloride NaCl, MgCl2, etc. Organic chemicals always write a two-dimensional view of molecules while representing the structure of the molecules. Example: benzene, naphthalene, etc. Physicists conjure up the three-dimensional views of molecular structures, while biologists describe the describe the protein structure at four levels. The sequence of amino acids, that is, the positional information in a protein, which is the first amino acid, which is second, and so on, is called the primary structure. Figure 9.3a. Primary structure of a protein. A protein is imagined as a line, the left end represented by the first amino acid and the right end represented by the last amino acid. A protein is imagined as line, the left end represents by the first amino acid and the right end represented by the last amino acid. The first amino acid is also called the N-terminal amino acid. The last amino acid is called the C-terminal amino acid. A protein thread does not exist throughout an extended rigid rod. The thread is folded in the form of a helix, similar to revolving staircase. Of course, only some proteins of the protein thread are arranged in the form of a helix. In proteins, only right-handed helices are observed. Other regions of the protein thread are folded into other forms in what is called the secondary structure. Figure 9.4.b. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to the tertiary structure. Figure 9.4c. This gives us three-dimensional view of a protein. Tertiary structure is absolutely necessary for the many biological activities of proteins. Some proteins are an assembly of more than one polypeptide or subunits. The manner in which these individual folded polypeptides or subunits are arranged with respect to each other. Example: linear string of spheres arranged one upon each other in the form of a cube or cube or a plate, etc. Is the architecture of a protein, otherwise called the quaternary structure of a protein. Adult human hemoglobin consists of four subunits. Two of these are identical to each other. Hence, two subunits of alpha type and two subunits of beta type together constitute the human hemoglobin Hb. Then, nature of bond linking monomers in a polymer. In a polypeptide or a protein, amino acids are linked by a peptide bond, which is formed when the carboxyl COH group of one amino acids react with the amino group of NH2 of the next amino acid with the elimination of water moiety. The process is called dehydration. In a polysaccharide, the individual monosaccharides are linked by a glycosidic bond. This bond is also formed by dehydration. This bond is formed between two carbon atoms of two adjacent monosaccharides. In a nucleic acid, a phosphate moiety links with the third carbon of one sugar of the nucleotide to the fifth carbon of the sugar of the succeeding nucleotide. The bond between the phosphate and the hydroxyl group of the sugar is an ester bond. As there is one such ester bond either side, it is called phosphodiester bond. Figure 9.5. Nucleic acids exhibit a wide variety of secondary structures. For example, one of the secondary structure exhibited by DNA is the famous Watson-Crick model. This model says that DNA exists as a double helix. The two strands of polynucleotides are antiparallel, that is, run in the opposite direction. The backbone is formed by the sugar-phosphate-sugar chain, and sugar chain. Sugar alternately located. sugar, phosphate, sugar chain. The nitrogen bases are projected more or less perpendicular to this backbone but face inside. The nitrogen bases are projected more or less perpendicular to this backbone but faces inside. And A and G, adenine and guanine of one strand compulsory base pairs with the thymine and cytosine respectively. On the other strand, there are two hydrogen bonds between adenine and thymine and three hydrogen bonds between G and C, guanine and cytosine. Each strand appears like a helical staircase. Each step of ascent is represented by a pair of bases. At each step of ascent, the strand turns 36 degrees. One full turn of the helical strand would involve 10 steps or 10 base pairs. Attempt drawing a line diagram. The pitch would be 34 Armstrong unit. The rise per the rise per base pair would be 3.4 Armstrong unit. This form of DNA with the above mentioned salient features is called bDNA. In higher classes, you will be told that there are more than a dozen forms of RNA, DNA named after English alphabets with unique structural features. Thank you.